Thank you so much. Thank you so much for coming. Thank you so much for coming, Brad, from UCLA. Would you like
Go
to
Bruins. Yeah.
No, go.
Go Bruins. It's nothing. Go ahead.
So I think we are very lucky also to have you both because obviously, Anne, you are the executive director of Ways Ancestor, and you are one of the organizations that is trying to sequence life on Earth under the umbrella of EBP, the Earth Biogenome Project. But Brad, you also have your own organization who is also sequencing life on Earth in California, precisely, right?
Correct.
And is also actively working as an affiliated project to the Earth Biogenome Project, right?
Correct. Yep.
Great, perfect. So first question to Anne. I think that I mentioned a lot, some information about the importance of integrating iPSCs into the scientific moonshot of sequencing life on Earth. But I bet the public would like to know more about why is it so important to integrate iPSCs into this important work?
That's a great question. Thank you, Alex. You laid it out nicely, the sad news that we're in the middle of a sixth mass extinction. It's actually the only mass extinction that is completely caused by human behavior. And so Wise Ancestors is looking at this problem in the sense that we feel like we need to use all the tools in the toolbox to actually address it. And so the two sides of addressing it that we are utilizing, number one is conservation genomics by creating tools for that form of conservation, namely generating reference genomes for endangered and culturally relevant species, but also biobanking samples, but also supporting local initiatives, indigenous communities in the work that they do. Why is that important? One of the main reasons we still have the existing biodiversity that is on this planet is because of, and thanks to, indigenous and local communities who know how to steward the land, that have been stewarding the land for millennia. So we have a lot to learn from these communities. It's basically the right thing to do. So an example I can share is a project that we have in Colombia, where it was a really nice, the beginning of the project was amazing. We're working with some researchers from the Humboldt Institute in Colombia who are working with local community members and a local NGO called Neotropical Innovation. And during the beginning of the work, they were doing these bird watching expeditions to count the number of species. And it was the local community members who noticed a bird song that they hadn't heard before. That kind of knowledge that this was something different led to a new species being discovered. The interesting thing is that species, they're not seeing very many individuals of this species. So it's possible that this one species is going to be rated as being critically endangered as soon as it gets a name. So this collaboration between the local scientists and the local community members led to the discovery of a new species. This project that we're funding funds the sequencing of that species, but it also funds the local community to be able to continue participate in the conservation. It's called the Biomonitors Program, and it involves funding 5 local community members for a year to be trained in genomic technologies and also to learn about how they can transition from the type of farming that the local community does right now in this region, which is largely monoculture and also a lot of cattle ranching, to transition that to more sustainable farming methods. And this particular project, they will be learning how to plant and raise a particular native blueberry plant that the birds live off of, and it is also useful for human consumption. So it's something that they can actually, the people that live in this area can have an economic benefit from. So this is a way that we're involving the local communities in the conservation of this particular region.
Thank you. Thank you for this enlightenment. I think it's making more sense. And yeah, explaining to people why is it so important to integrate IPSCs to this endeavor. Brad, you are the director of the California Conservation Genomic Project, and you have been sequenced a lot of different species here. Would you be able to walk us through your approach?
Sure. So first of all, as Alex said, I'm a professor at UCLA, and I have a joint appointment in 2 departments, ecology and evolutionary biology, and in a very integrative group called the Institute of the Environment and Sustainability. And the project that we're talking about is one of the main projects that I've been running for the last several years, called the California Conservation Genomics Project. And the idea behind it is really very, very simple, which is that if there is a single rule of certainly evolutionary biology, and biology perhaps in general, it is that variation is good. Variation allows you to respond to different challenges, variation at the individual level, variation at the population level, variation at the species level. And we all know the flip side of that, which is that inbreeding is bad. You know, you express diseases, individuals are sort of genetically weakened by inbreeding, and the opposite of that is outbreeding, and it's the consequence of outbreeding is increased genetic variation. Well, it is clear, as we have heard, that a lot of species are going extinct. By some estimates, during the course of my son's lifespan, my son will be 35 next week. In the course of his lifespan, we may lose 1 million out of those roughly 2 million described species. How many undescribed ones we lose is anybody's guess. It's a lot of species. And even if that estimate's off by an order of magnitude, 100,000 is simply too many. It's just unacceptable. So the question is, how do we deal with that? How do we figure out a way to cut down that loss? We're not going to eliminate it, but how can we reduce it? And the idea behind the California Conservation Genomics Project is to say, well, if we can do rapid genomic inventories of a lot of species who are co-distributed across a landscape, what we can do is we can identify potentially parts of a landscape that harbor genetic variation, that harbor a lot of it. Places where populations are not and individuals are not inbred, but they're outbred. And do that across oak trees and California poppies and gopher snakes and damselflies and butterflies, across a whole wide range of taxa. And if we can do that and protect those landscapes, then what? Then, as we humans continue to change climate, as we continue to add environmental insults to California or to any other place, we've protected the populations that have the best chance of evolving their way out of that crisis. They have the most standing genetic variation. They have the most capacity to be able to evolve their way out. Maybe they will, maybe they won't, but they're our best shot. And when you're talking about hundreds of thousands of species potentially going down the drain, that's a very, very, very powerful tool. So with the California Conservation Genomics Project, what we've done is brought together 77 different investigators from across the 10 University of California campuses to identify and contribute samples of about 250 species of plants, of invertebrates, of vertebrates, of inshore marine taxa. And for each one of those species, contribute about 150 individuals tissue samples spread across the state that we are then, under the project, sequencing at the whole genome level at high coverage so that we really understand genetic variation across all of those species, across all of that landscape of California, and use that in order to identify the landscapes that are the most important for climate resilience and biological resilience, make sure that those landscapes are connected by reasonable biological corridors, and that they will at least give us a shot at cutting back on some of this biodiversity crisis. And we're just about done with the project. We anticipate having the final data collected in a month or two, and being able to wrap this up and deliver it to the legislature and other regulatory bodies, U.S. Fish and Wildlife Service, California Department of Fish and Wildlife, a lot of groups that we work with very, very closely, by the end of the summer. So that's the project.
Ooh, congratulations.
Thank you.
Really. Since we are here talking about fundings also, may I ask to you and Brad, where are coming those fundings? Because yeah, maybe it wasn't clear enough. All those data generated are open sourced, right?
Sorry, say that again?
All the data generated are open sourced, right?
Correct.
So who is paying for this work, tremendous work?
In my case, you are. The state of California is paying for it, and inspired member of the state legislature named Richard Bloom from Southern California, from LA, partnered with us at UCLA and other UC campuses to build a consortium of members of the state legislature. This was back in 2019, or 2018, excuse me. We got that written, we got $12 million written into the state budget, which by some miracle survived the state budgetary process. And so that funding has gone to create this new set of about 21,000 fully resequenced genomes across the state of California, as well as all the infrastructure for analyzing the data, storing the data, and delivering it in a consumable way for anybody from a biologist like myself to a member of the state legislature.
So in our case, I would say we're probably the opposite.
There you go.
The other extreme of how to get this kind of funding. We're a nonprofit. We post conservation challenges on our platform. These are decentralized. It's DeSci, basically, as Alex explained. And part of the function of the platform is to crowdfund for the project. So people can come on the website and look at all of the different projects on our dashboard. They can see how much a particular project is going to cost, how much has been raised so far, and they can contribute to it that way. And we also apply for grant funding. Sometimes there will be a foundation or some sort of organization that wants to fund for conservation of a certain species, so we'll apply for grant funding in that case. But it's very much decentralized in the sense that we invite the community to help us complete these projects.
Okay, thank you. And continuing on this thread, so both of your projects, and I'm part of one of them, both of those projects are under the umbrella of EBP, which is the Earth Biogenome Project. And Earth Biogenome Project have like a very interesting decentralized method of like completing the moonshot of sequencing all life on Earth, which is like having multiple affiliated projects, one in California, another one working with IPSCs. There is like, I don't know how many projects are under EBP right now, but a lot, right? But it's very under 60?
Just about 60.
60. Yeah. And so what do you think about this decentralized methods of EBP?
So I'm very involved with the EBP. I've been a part of the kind of central group for quite a while with EBP. I think I guess there's two things I would say about EBP. First of all, it's the Earth Biogenome Project, and what the EBP seeks to do, as you said, is to sequence one individual and produce a very good reference genome, it's called, so a very complete chromosome-level genome, ideally for one individual of every species of the 1.8 million species described on Earth. That's a tremendous resource. In and of itself, though, it doesn't, at least it helps inform evolutionary biology, and I'm also an evolutionary biologist. It only peripherally helps inform conservation, because conservation is not about understanding a single individual. It's about understanding populations. It's about understanding population and species health and resilience and declines. And for that, you do need more. It's a very, very important first step. In terms of the funding model for EBP, I think it's fair to say it's been a real challenge. What EBP is a hub for projects like mine, like Anne's, like many, many others around the world. The Biodiversity Genomics Europe Genomics project, other projects like that. And I think that where EBP needs help is with actually having some funding. However that occurs is up for grabs, but it's been a challenge.
Do you think that EBP, so Earth Biogenome Project, should also create an Earth Biogenome Fund?
You want to take that?
I would say that would be great for the EBP, but what I would love to see happen is a change in people's mindset about funding the commons, and the commons in this case being biodiversity and the data that comes from it, because we're not really limited by the technology. We're not so much limited by figuring out what to do to save biodiversity, but we need the input of resources, of funding resources. So in the case of Wise Ancestors, we're just one organization, but we could actually make a dent because we've set up a model where we can have an unlimited number of projects happening in parallel on our dashboard that are funded by people, individuals. Just if everybody just gave a little bit, we could get data and involve local communities wherever these species are all over the world for not a whole lot of money from individual people. So I think that's another way to go. So there's room for both. Maybe governments should all fund an EBP fund, similar to what's happening at the UN level with the Convention on Biological Diversity. Perhaps that should exist, but I think also organizations like ours can make a big difference if we can just get people to realize this is a real problem and it needs to be addressed.
And larger question, Brad, I have you on stage, so I can ask this question. More largely, what do you think would be the best solution to the biodiversity crisis?
Yeah, that's a nice, easy question, right? What would be the best solution to the biodiversity crisis? Well, one can think a little bit ultimately, one can think a little bit proximately. Ultimately, in my view, and this may be a place where Anne and I disagree to some extent, but ultimately, or perhaps not, for biodiversity, people are the problem. And the best solution to the biodiversity crisis would be, at some level, to go back to the 1960s and ZPG, right? Zero population growth or decline at this point, and reduce the human footprint on the planet. There's different ways one can do that, but ultimately, if we're not going to completely take over all the space of the planet, that's kind of what we have to do, right? We have to reduce our footprint. And we can reduce our footprint by, being a vegetarian and not driving and doing all the things that I'm sure all three of us do. But that's kind of small change. The bigger one is to simply have fewer people impacting the planet. I think at the more proximate level, I think the best thing we can do to at least slow the biodiversity crisis is, again, I think at a global scale or at a national scale, bringing in any and all partners who we possibly can. We need to identify the healthiest populations, the most resilient populations of the largest number of species, including common species, including those species that we take for granted, because, everything is less common than it used to be. And identify those species and those taxa and those regions where we can protect those areas, save them for posterity, and allow them, as I said, to sort of work out their community structure and their evolutionary future in a way that allows them to survive.
I 100% do agree with you, Brad. Don't know how I gave you the impression I wouldn't, but adding to that, I would say we need to shift human behavior. And so projects where it was mentioned earlier, just seeding local projects where the behavior is more sustainable, where people only take what they need, not overforesting, not overharvesting, still using the environment for a bioeconomy. We do need to live off of the land, but making that happen in pockets around the world to start to shift the behavior, I think, needs to happen.
Thank you so much to both of you. Thank you.
Thank you.
Thanks, Alex.